Hello everyone. Today we are going to solve NCERT exercise questions of chapter number 5, that is molecular basis of inheritance. So here's the disclaimer. What are going to be today's learning objectives? So today what we are going to do, we are going to solve the NCERT exercise questions of chapter number 5, that is molecular basis of inheritance. And what we will do by solving these questions, so when we were solving this question during that period, we are going to understand and explore more fundamental concept of this chapter number 5 that we have studied in previous classes. But by solving these questions, these NCERT questions, we are going to understand it in more details. Okay, in more details as in like in more depth. And if there was any confusion, if you guys have any confusion during the classes and during any concept, maybe solving these questions clear that doubts. Then today's index. So today we are going to solve question number 1 to question number 14 of NCERT exercises. Okay, for which chapter? For chapter number 5, that is molecular basis of inheritance. Now let's start the question answer. So first question, this is NCERT exercise first question, and the question says group the following as nitrogenous bases and nucleosides: adenine, cytidine, thymine, guanosine, uracil, and cytosine. This is from NCERT section 5.1.1. So basically in this question what they are asking, they have given us some options, some 6 options, and now they are telling us among these 6 options we have to choose which are nitrogenous bases and which are nucleosides. What are nucleosides? Nucleosides is a molecule composed of nitrogenous base linked to a pentose sugar. Okay, that pentose sugar could be ribose or deoxyribose depending on RNA or DNA, what we have to So nucleoside that, and if we are going to add one phosphate group, so what it will become? It will become nucleotide. But here they have asked us about the among them which are nitrogenous bases and which are nucleosides. So talking about nitrogenous bases among these options are adenine, thymine, uracil, cytosine, whereas the nucleosides are cytidine and guanosine. So here are the pictorial representation of these nitrogenous bases as well as nucleosides. Next question. So next question says what? Let's see. This is NCERT exercise question number 2, and this says if a double-stranded DNA has 20% of cytosine, calculate the percent of adenine in the DNA. This is from NCERT section 5.1.1. So the question says, so there is a double-stranded DNA and it has 20% of cytosine in it. Now we have to calculate the percentage of adenine in that DNA. Let's see how we can solve this question. This is kind of a numerical question. Let's see how we will solve it. So we can solve it by using Chargaff's rule. Who proposed Chargaff's rule? So it was proposed by Erwin Chargaff for a double-stranded DNA. So what this says the DNA molecule should have an equal ratio of pyrimidine, that is cytosine and thymine, and here we are talking about DNA only. Okay, so cytosine and thymine, and purine, which is adenine and guanine. It means what? It means that the number of adenine molecule is going to be equal to thymine molecules, and the number of guanine molecules is going to equal to what molecules? Cytosine molecules. So because we know that they have to do what? They have to complement each other. So definitely if there are 20 number of adenine, so there are going to be 20 number of thymine also, because they are going to be together. Okay, there will be a base pairing among them. Same ways for guanine and cytosine, we know that guanine, if it is like 80 guanine, then there will be 80 cytosine also. We know that because they have to Base pair means they have to complement each other. They are complementary to each other. Now, so let's solve the questions. According to this Chargaff's rule, if double-stranded DNA has 20% of the cytosine, then according to the law, it would have how much percentage of guanine? It's going to have 20% of guanine only. So the percentage of cytosine and guanine will be how much? 20 plus 20, that is 40%. The remaining will be represent what? A plus T molecule. Since adenine and thymine are always present in equal number, so the percentage of adenine molecule will be how much? 60 divided by 2, that will be 30%. So the answer of this question is that the percentage of adenine molecule in this DNA is what? It's 30%. So if we will look at the sum total of all, so there will be 20% of cytosine, 20% of the guanine, 30% of adenine and 30% of thymine. And if you will sum it all, you will see it will be like 100% only. Now, let's come to the next question. Next question says, this is NCERT exercise 3. Question is very simple. They have given us a sequence of one strand of DNA. So this says, if the sequence of one strand of DNA is written as follow. So they have given a strand from which direction? 5' to 3' direction. But now let's see the question. What is the question? So question is, write down the sequence of complementary strand in 5' to 3' direction. They have asked us to write the sequence of the complementary strands. So this 5' to 3' is going to make whatever complementary strand, we have to give the sequence of that complementary strand, but in which direction? In 5' to 3' direction. And this is from NCERT section 5.1.1. So how they will complement, like how will this complementary strand will be made? This complementary base pairing, the DNA strands are complementary to each other with respect to base sequence. Adenine going to pairs with thymine via 2 hydrogen bonds, whereas guanine going to pair with cytosine via 3 hydrogen bonds. Okay. So now what will happen? If the sequence of one strand of DNA is 5' to 3', if this sequence, the given sequence is like this, so what is going to be the sequence of complementary strand? It will be like this, 3' to 5. It will be in the direction of 3' to 5'. And how will it complement? Adenine complements like what? And will become like what? T. T complements with A, G with C, C with G, A with T, T with A, G with C, C with G, A with T, T with A, G with C, C with G, A with T, T with A, G with C, C with G, A with T, T with A, G with C, C with G, A with T, T with A, G with C, C with G, A with T, T with A, G with C, and last C with G. So this is the complementary strand of this sequence, okay, of this given sequence. But now this is in which direction? This is in the direction of 3' to 5'. But they have asked us in which direction? So they have asked the sequence of the complementary strands in 5' to 3' direction, and therefore the sequence of nucleotides on DNA polypeptide on the complementary strands in 5' to 3' direction is this. It is like they did what? They just reversed it. You guys can see, if you will read it, like it will be like reverse. 5' to 3', it will be like GCAT, GCA, like this. It is like this. Okay. So this is a very simple question. Now let's go to next question. Next question is NCERT exercise question number 4. Question says what? Question says, if the sequence of the coding strand in a transcription unit is written as follows. So they have given us again a sequence, but here now the sequence is of coding strand and where? In a transcription unit. Okay, now we have to write down the sequence of mRNA, and this is NCERT. This is from NCERT section 5.5.1. So let's see the answer. So what's the trick here? So here the trick is that, not a trick, this is we know that coding strand is what? Coding strand is the sequence of the DNA that has the same base sequence as that of mRNA, because we know that in the transcription unit this coding strand is not going to transcribe, irrespective of the fact that the strand name is coding strand, but the strand which is going to transcribe into mRNA is what? is the template strand. But because this is complementary to this template strand which is going to transcribe, that's why this coding strand sequence, DNA sequence, has the same base sequence as that of mRNA, but only with one difference. With what difference? This mRNA will have uracil instead of thymine. Okay. So this is the coding strand in the transcription unit 5' to 3'. So what will be the RNA? Everything will be same, but thymine is replaced by uracil. Hence the sequence of mRNA will be, here you guys can see everything is same, but wherever there is thymine, it is replaced by uracil. Here you guys can see AATU, it got replaced. GGCCAATU, 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 GC A, again U, again G, again C, again A, again U, because there is presence of, it is like here thymine, then it will be uracil, then again G, again C, again A, but again uracil instead of thymine, again G and again C. So this is the sequence of mRNA. Now comes to next question, NCERT exercise question number 5, which question says, which property of DNA double helix led Watson and Crick to hypothesize semi-conservative mode of DNA replication? Explain. So this is from NCERT session 5.4. So this question says that what are the properties of this DNA double helix structure, okay, that made Watson and Crick to give a hypothesis that the DNA replication mode is what? Semi-conservative in nature. So let's see the answer. What will be the properties of that DNA helix? Let's see. So the key property of the DNA double helix that led Watson and Crick to propose the semi-conservative mode of replication was, first, complementary base pairing between two DNA strands. So we know that between these two DNA strands, but what is present, like what is there? There has always been a complementary base pairing. Means purine is going to complement with pyrimidine. Okay, here also we guys can see that G pairs up with C. This again CG, AT. So it will be like this base pairing. DNA consists of two strands that are twisted around each other to form what? To form double helix. These strands are held together by hydrogen bonds between specific pair of nitrogenous base. Adenine always pairs with thymine and guanine always pair with cytosine in DNA replication. So here again, this DNA double helix, we can see what is there. There are two strands that are twisted around each other to form double helix, and these strands are held together by what? By the hydrogen bonds between the specific pair of nitrogenous base. Adenine and thymine pairs with help of two hydrogen bonds, whereas guanine and cytosine pairs up with three hydrogen bonds. Okay. Now, next is antiparallel strands. The two strands of DNA run in opposite directions, meaning they are antiparallel. This arrangement is crucial for complementary base pairing to occur. So we know that in DNA strands, if one is running from 5' to 3' direction, okay, see 5' to 3' direction, the other will be running from 3' to 5' direction. Okay, so these are antiparallel strands. These run in different direction. Now, if we talk about this semi-conservative replication, Watson and Crick understood that two strands of DNA are what? Complementary. They also recognized that these strands run in opposite directions. This led them to a reason that during replication, these two strands could separate and each could act as what? Each could act as template. So here we can see what happened. The two strands separated, each is acting as a template and the replication is happening. Now, resulting DNA molecules, if we talk about, this process would result in two new DNA molecules, each composed of original parental strand and one newly synthesized strand. This is the semi-conservative model of DNA replication, where each new DNA molecules retain half of the original DNA and then half is newly synthesized. So this is the what? Semi-conservative nature of this DNA replication. And this is what Watson and Crick has hypothesized. Now let's come to next question. This is NCERT exercise question number 6. So what this question is saying, let's see. This question is saying us, depending upon the chemical nature of the templates, that what is the chemical nature of that template, is it DNA or RNA, and the nature of the nucleic acid synthesized from it. So like what kind of nucleic acid is going to synthesize from that template, whether it is DNA or RNA, list the type of nucleic acid polymerases. So we know that in this chapter, we have studied about 2 polymerases. Let's see. First is DNA dependent DNA polymerase. So why we are calling it DNA dependent? Because this is the DNA is the template. And DNA polymerase, why? Because this is the DNA is the nucleic acid which is synthesized from it. Okay, so DNA dependent DNA polymerases. It's use a DNA template for synthesizing a new strand of DNA, and it replicates the DNA molecules, and this DNA dependent DNA polymerase comes into function during replication. So by it use a DNA template, and using that DNA template it synthesizes a new strand of DNA. Okay, it replicates the DNA molecule. So here is this DNA replication has been shown. Here is what? Here is the DNA polymerase, and we know what it can do. It can add the nucleotide to the newly synthesizing strand. And in which direction? 5' to 3' direction they can add. Now, going to second type of this nucleic acid polymerase, it is DNA dependent RNA polymerase. Why DNA dependent? Because the nature of the template here is the DNA, whereas the nucleic acid which is going to synthesize from it is RNA. So that's why we are going to say that this is DNA dependent RNA polymerase. It comes into function during what? It comes into function during transcription. But its function, it's use a DNA template strand for synthesizing what? For synthesizing RNA. It synthesizes the RNA during transcription. Here you guys can see it's what? This is the double-stranded DNA. This is the RNA polymerase, and with the help of this template strand, this RNA has been synthesized. So these are the 2 different polymerases that we have studied in this chapter. Now comes the NCERT exercise question number 7. The question says, how did Hershey and Chase differentiate between DNA and protein in their experiment while proving that DNA is the genetic material? This is from NCERT session 5.2.1. So now what they are asking us, this question is telling us, we have to tell that how this Hershey and Chase. So this Hershey and Chase were the scientists, and we have studied about their experiment, what they have, how they have proved that DNA is the genetic material. Their experiment, if you guys remember, said the unequivocal experiment which has proved that DNA is the genetic material. So how they have differentiated between this DNA and protein in their experiment, we have to answer that question in this. We have to answer that in this question, and this is from NCERT session 5.2.1. So let's get started. So in this question, we are going to study about that what make them to differentiate between DNA and protein in their experiment. So Hershey and Chase used radioactive isotopes to differentiate between DNA and protein in their experiment. Used different radioactive isotopes. So what radioactive isotopes they have used? They used this radioactive phosphorus 32P to label DNA, and 35S, that is radioactive sulfur, to label proteins. DNA labeling they did how? DNA which contains DNA we know contains phosphorus but not sulfur. They grew bacteriophage in a medium which containing radioactive phosphorus. This resulted in phage DNA being labeled with radioactive phosphorus. Whereas if we talk about the protein labeling, protein contained what? Sulfur but not phosphorus. They grew a separate batch of bacteriophage in a medium containing what? Radioactive sulfur. This result in phage protein coat being labeled with radioactive sulfur. So actually, if you guys remember what happened in that experiment, so this is what? This separate batch of bacteriophage, it has been grown in medium which contain what? Radioactive sulfur. And now what will it result in? It will result in that this phage protein coat will being labeled with radioactive sulfur. Whereas in this batch of bacteriophage, it has been grown in a medium containing radioactive phosphorus. Therefore, what is happening in this bacteriophage? In this batch of bacteriophage, DNA were being labeled with phosphorus, radioactive phosphorus. So now what they did, then they infected this bacteria with this bacteriophage. Okay, after infection, they did this blending step, which is the removal of this protein coat from the surface of bacteria. Then third, they did the centrifugation, and what the result of centrifugation has come. So in the batch of this bacteriophage, which has been grown in radioactive sulfur, no radioactivity was detected in the cells, whereas the radioactive were detected in supernatant. Whereas in the other batch, who were grown in the medium which is containing the radioactive phosphorus, radioactivity has been detected in the cells, but no radioactivity has been detected in the supernatant, and this proves that the DNA is the actual genetic material. Now let's come to NCERT exercise question number 8. So what this question is saying us, so it is a simple question. They have given us terms, and now they are telling us we have to differentiate between the following. First is the repetitive DNA and satellite DNA. This is from NCERT session 5.9 and 5.10. Then there is we have to differentiate between mRNA and tRNA. This is from NCERT session 5.9 and 5.6.2. Then third, there is that we have to differentiate between template strand and coding strand. This is from NCERT session 5.5.1. So let's start with the first differentiation, that is between repetitive DNA and satellite DNA. So if you will see what is the difference between the repetitive DNA and satellite DNA. So this repetitive DNA is the DNA sequences that are repeated multiple times within genome. Okay, whereas this satellite DNA is what? It is a specific type of repetitive DNA, and it is characterized by what? It is characterized by the long stretches of highly repetitive sequence. Okay, now this repetitive DNA can be found throughout the genome, whereas this satellite DNA is concentrated at specific location, for example centromeres and telomeres. This repetitive sequence, they have moderately repetitive sequence. They have, as I said, 100 to 105 repeats per genome. But this satellite DNA, this consists of highly repetitive sequence, even more than 106 repeats per genome. Okay. Now, if we talk about mRNA and tRNA, differentiate between mRNA and tRNA. So this mRNA, or what we call messenger RNA, carries the genetic information from DNA to the ribosome, acting as a template for protein synthesis. Okay. Whereas what is a tRNA? tRNA is a transfer RNA which acts as an adapter molecule, and its function is that it carries a specific amino acid to mRNA for the synthesis of polypeptide. This mRNA, this is linear molecule, much longer than tRNA. But if we talk about the tRNA, if it's 2D dimensional, we would talk about it is a clover leaf shaped molecules with a unique anticodon loop. So what this anticodon loop has? Anticodon loop has nucleotides which are complement to the codon which are present on the specific codon which are present on the mRNA. So this is the structure of mature mRNA. What is this? This is the start site. Here there will be codon of AUG. AUG is what? It is start codon, as well as it codes for methionine. There will be any stop codon. Okay, stop site. These are what? These are the untranslated region. 3' and 5' and 3' untranslated region. So these are the regions which are not going to be translated. These are untranslated region, but they help in increase the efficiency of translation. And this 5' capping, okay, with that methylated guanine triphosphate, and then this 3' poly adenylated tail. Okay, this is the structure of a mature mRNA. But if we talk about the clover leaf of tRNA, it is looking like this. This is the anticodon loop, where there will be nucleotide that is complementary to the codon present on the mRNA, as well as it has an acceptor arm where the amino acid will come and attach. Now let's come to the third differentiation, that is the template strand and the coding strand. So what is the template strand and coding strand? This we are talking in the terms of transcription unit. Okay, so the template strand is the DNA strand that is read by RNA polymerase during this transcription to create what? To create a complementary RNA molecule. Whereas what is a coding strand? Coding strand is a sequence of the DNA that has same base sequence as that of mRNA, except thymine that is replaced by uracil in RNA. And this is the same question that we have. This is the same topic from which we have done a question. I think it was second or third question. Okay. If we talk about the template strand, it runs from 3' to 5', but if we talk about coding strand, it runs from 5' to 3'. This name is like confuse you because it is like coding strand, but it is not coding for this RNA molecule strand, RNA molecule. Okay, this has the sequence of DNA that has same base sequence as that of mRNA, but it is not going. This is not the strand which is going to be transcribed. It is the template strand which is going to be transcribed during transcription, and it runs from 3' to 5', and this coding strand runs from 5' to 3'. So this is the difference between template strand and coding strand. Now we come to NCERT exercise 9. Okay, so what's the question today? So question here, so the question is, list 2 essential role of ribosome during translation. So we have to tell the 2 essential role that ribosome plays during the translation. Translation is the process where the mRNA is translated into what? Into protein. This is from NCERT session 5.7. So 2 essential roles of ribosome during translation. First, providing the site for protein synthesis. It is providing what? It is providing the site for protein synthesis. Ribosome consists of 2 subunits, a small subunit and a large subunit. The small subunit attaches to mRNA, whereas the large subunit holds the tRNA and nascent peptide. This arrangement ensures the correct order of amino acid is incorporated into proteins guided by mRNA sequence. So what it is trying to tell us? So it is going to trying to tell us that first of all, this ribosome is providing site for protein synthesis. How? This ribosome is consisting of 2 subunits. So we have talked about that ribosome has 2 subunits, one smaller and larger, and then other is larger. Okay, so this smaller subunit goes and attached to the mRNA that you guys can even see in the picture, that the small subunit came and bind to the mRNA, while this large subunit holds the tRNA and the nascent peptide. Okay, and this is the arrangement that will make sure that the correct order of amino acid has been incorporated into the protein, which is guided by what? Which is guided by this mRNA sequence. So first role is this. Now what is the second role? So the second role is it's catalyzing the formation of peptide bonds. So this large subunit of ribosome contains an active site, okay, where these peptide bonds are formed between adjacent amino acid. This process, catalyzed by ribosomal RNA, links the amino acid together to form polypeptide chain. The ribosome ensures the proper orientation and proximity of the amino acid for this reaction to occur efficiently. So first we have talked about the, it providing us the site for protein synthesis. Second is that it also catalyzing the formation of the peptide bond. We know that the 2 amino acid are going to be bind together with a peptide bond. So the larger subunit of this ribosome contain an active site where this peptide bond formation take place between the 2 adjacent amino acid. This process is catalyzed by a ribosomal RNA, which links this amino acid together to create this polypeptide chain. And the ribosome also ensures the proper orientation and proximity of the amino acid for this reaction to occur efficiently. So these are the 2 roles which ribosome plays during translation. Now NCERT exercise 10. So question is that in the medium where E. coli was growing, lactose was added. Okay, we know that lactose act as inducer. When which induce the lac operon. Then why does lac operon shut down sometime after addition of lactose in the medium? This is from NCERT section 5.8.1. So the question is saying us The question is saying us that, in a medium where this E. coli was growing, they added the lactose. Okay, we know that lactose acts as an inducer, and which induce this lac operon. Okay, but after some time, this lac operon shut down. After this addition of lactose in the medium, after some time, this lac operon shut down. Now we have to answer why this happened. Let's see the answer. If you guys see the answer is that lac operon shut down sometime after addition of lactose in medium because of the following reason. So we are going to see the sequential step what is going to happen. Okay, first what will happen? Lactose induction. When lactose is present in the medium, it enters the E. coli cell. It is then converted to allolactose, which act as inducer. So what happens in this? In this lactose induction, what happens? When this lactose is present in the medium, okay, it enters the E. coli cell. Now, this lactose will get converted to allolactose, and which act as an inducer. So, you guys can see if this inducer, if this allolactose is present, it will go and bind to the repressor protein. Now, this repressor protein become what? This repressor protein become inactive. Now, because this is inactive, it is not going to bind to the operator. And it is not binding to the operator, allowing the RNA polymerase to bind to the promoter. And once the RNA polymerase will bind to the promoter, transcription will take place. After that, what will happen? This lac mRNA will be formed, and this will be translated to the proteins. So this is the normal step that's going to happen. As I said, repressor inactivation will happen. Allolactose bind to the lac repressor proteins, preventing it from binding to the operator site on the DNA. Okay. Then what will happen? Transcription begins. With the inactive repressor, RNA polymerase can bind to where? Promoter region. It allows to initiate transcription of the lac operon genes, which are responsible for what? These are responsible for the lactose metabolism. So now here we know what happened. Okay, the transcription begin, and then there will be the production of the proteins which are responsible for lactose metabolism. Now what will happen? The enzyme which is produced by lac operon, primarily which enzyme? Beta galactosidase. This beta galactosidase is going to break down lactose into sugar and into glucose and galactose. Okay. Now what will happen? Because this has already break down lactose is consumed, its concentration decreases, and it allows what? Now, because this concentration decreases, leading to the decrease in low lactose level also, the repressor protein become active again and bind to where? Bind to operator site. Now, what will happen? Operon shutdown will happen because the binding of the repressor to the operator blocks RNA polymerase. This halt transcription of lac operon genes, and effectively it will shut down the operon. So now what will happen? Because of this breakdown of the lactose, because of the beta galactosidase enzyme, now there is depletion of lactose. Now because lactose is consumed, okay, its concentration got decreased, it will lead to decrease in also allolactose level. Okay, now there is no inducer which is going to now bind to this repressor protein. So now what will happen? This repressor will go and bind to operator region, and now this will do what? It will prevent RNA polymerase from RNA polymerase to bind to the promoter site, and what will happen? This halt transcription of the lac operon genes, and effectively it will shut down the operon. So it is doing what? It is preventing mRNA polymerase from transcribing this operon. So this is how it will stop after some time of adding lactose in the media. Now, NCERT exercise 11, question number 11 means, question says, explain in 1 or 2 lines the function of the following. They have given us 3 terms and we have to tell the function of those things. First is promoter. This is from NCERT session 5.8.1. Then is tRNA, that is transfer RNA. It is NCERT session 5.6.2. Then it is exon, which is the NCERT session 5.5.2. So now we have to answer these. We have to tell about these in lines. Now, first comes promoter. Promoter is a region of DNA that help in initiating the process of transcription. It serves as a binding site for RNA polymerases. So, what is happening here. So if you guys can see what actually the promoter is a DNA, is a region of DNA, okay, that helps in what? That helps in initiating the process of transcription. It allows what? It allows the binding of RNA polymerase. Okay, and RNA polymerase with the help of the sigma factor will go and bind to this promoter site, and this will initiate the transcription. So this is what promoter is. Now, next we come to tRNA. What is a tRNA? tRNA, also known as a transfer RNA, is a adapter molecule, is a small RNA that reads the genetic code present on mRNA. It carries the specific amino acid to mRNA on ribosome during translation of protein. So what is the function of this tRNA? It is doing what? It is a small RNA that is reading the genetic code on the mRNA with the help of this anticodon loop. Okay, and it carries the specific amino acid to mRNA on ribosome during translation of protein. So during the translation of protein, it is doing what? It is reading the genetic code present on mRNA, as well as it is carrying the specific amino acid to mRNA on ribosome during translation of protein. So these are these function. Then comes exons. What are exons? Exons are coding sequence of DNA in eukaryote that is going to transcribe for proteins. The sequence of exons appear in mature processed RNA and are translated into proteins. So we know that when the DNA is transcribed into RNA, it is not in a mature form. mRNA is not in a mature form. It has intron as well as exon. Intron is like coding region, whereas intron is not. So we have to get rid of that intron. So with the help of splicing, we can get this intron removed. Once this intron got removed, we can use it as, now this mature and this capping and this tailing thing happens, and this, like this capping and tailing thing is present, and this as whole is called what? Like is the now mature processed RNA, which is going to translate into protein. Now, NCERT exercise question number 12. Question says, why is Human Genome Project called a mega project? This is from NCERT session 5.9. So the question is very simple. We have to just answer why this Human Genome Project is called a mega project, such a big project it is. Its duration is 13 years, started in 1990 and completed in 2003. Its main goal is to sequence all base pairs in human genome. If we talk about scale and cost, so human's genome size is what? It is approximately 3 into 10 to the raised power 9 base pairs. Now, estimated sequencing cost initially is $3 per base pairs, and if we will total estimate the cost, it will become approximately $9 billion US. So this is the Human Genome Project scale and cost. Then, if we talk about data storage challenge, so storing this genome sequence in the book as type form. So if we want to store this genome sequence in book as type form, so how much books we required? We required approximately 3,300 books to store the data from one human cell, and in that each 3,300 books there should be 1,000 pages per book, and in that book, 1,000 pages book, there should be 100 letters per page. So you can understand like how much big the data is. Now, if we talk about the computational needs, enormous data volume necessitated the high-speed computational devices. It is essential for data storage, retrieval, and analysis. Okay, so it is like enormous data volume which needed high-speed computational device, and it is essential for data storage, retrieval, and analysis. Now, let's come to next question, which is NCERT exercise question number, means for NCERT exercise, it is question number 13. Question says, what is DNA fingerprinting? And we have to mention its application. Okay, and this is from NCERT session 5.10. Let's start. So what is DNA fingerprinting? So if we talk about the DNA fingerprinting, it is a technique which is used to identify and analyze the variation in various individuals at the level of DNA. So it is a technique which used to identify and analyze the variation in various individuals at the level of DNA. It is based on what? It is based on the repetitive DNA, which is the variability and polymorphism in DNA sequence. So this is the schematic representation of DNA fingerprinting that we have studied in class. I think yesterday's class only, like last class only. Then, what are the application of this DNA fingerprinting? We can use in crime investigation. It helps to identify criminals, connecting suspect to crime scene. For example, in this figure we can see, so this is the evidence they have collected from the crime scene, and when they have performed this DNA fingerprinting, they have found it is matching to suspect number B. So like this we can use this DNA fingerprinting. Then we can use it in where? We can use it in family matters. It helps to prove parentage or testing of paternity. It helps to resolve family dispute and immigration cases. Okay, this is the DNA fingerprint profile of child and parents. Now we come to the last question of NCERT exercises. Okay, this is the question number 14, and what this question says. So in this question we have to briefly describe the following. First we have to describe the transcription. This is from NCERT section 5.5. Then we have to describe polymorphism, which is from NCERT section 5.10. Then translation from NCERT section 5.7. Last bioinformatives, which is NCERT section 5.10. So let's see the answer. So if we talk about transcription, what is a transcription? It is a process of copying genetic information from one strand of DNA into RNA. So what is the process of this transcription? If we define this process of transcription, what is a transcription? It is a process in which there has been copying of genetic information from one strand of RNA into from one strand of DNA into RNA. So what are the requirements? First key enzyme is RNA polymerase enzyme. What kind of RNA polymerase? DNA dependent RNA polymerase enzyme. Then we need DNA template. Then we need 4 types of ribonucleotides. Okay. Now, what are the different steps of this transcription? So there are 3 main steps of this transcription. First step is initiation. So what happened in this initiation step? RNA polymerase bind to promoter region to initiate. It requires sigma factor for initiation. So what happened in this transcription process? So RNA polymerase, which has this sigma factor, it requires sigma factor to bind to what? To bind to promoter region. And once it got bind to promoter region, it will initiate the transcription. Then after initiation step, which step will come? The step will come elongation. What happened in this elongation? RNA polymerase use the nucleoside triphosphate as substrate. It polymerizes in a template dependent fashion following the principle of complementarity. Here you guys can see what is happening. So this, after the initiation process, this elongation, this means after this initiation step, what happened? RNA polymerase go and bind to the promoter region, and this transcription initiated, it started. So now what happened in this elongation step? In elongation step, this RNA polymerase used this nucleoside triphosphate as substrate, and it polymerizes in template dependent fashion only, and it follows what? Principle of complementarity only. Means purine going to complement with pyrimidine. But here the only difference is that this adenine will go and complement with uracil. Okay, not thymine, because we know that uracil is present in mRNA. Okay, we know that the DNA dependent RNA polymerase can add nucleotide in the direction of 5' to 3' only. So we know that from which strand this means RNA this will transcribe from 3' to 5' strand, which is the template strand. Okay. Then the last stage is termination. Once the polymerase reaches the terminator region, the nascent RNA falls off. This is also because the RNA polymerase detach. This termination process requires what? This termination process will require rho factor. So at last what will happen? This, with the help of this rho factor, this polymerase reaches the termination regions and the nascent RNA falls off. this causes the RNA polymerase to detach. Okay, so this is the process of, these are the main three stages of transcription. Then comes polymorphism. So definition of polymorphism is what? It is the existence of different nucleotide sequence, okay, at a specific DNA site within a population because of mutation in DNA. So this is the variation at the specific DNA site within a population because of mutation in DNA. And if this mutation is heritable, okay, and is observed in population at frequency greater than 1%, then this will consider DNA polymorphism. What is the origin of polymorphism? So this polymorphism arises due to mutation in DNA sequences. Mutation can occur in somatic cell or germ cells. These mutations are maintained through inheritance, and inherited mutations accumulate over generation, which contribute to this variability and polymorphism. Okay. Now, if we talk about the significance of this polymorphism, so what are the significance of this polymorphism? So it will create the genetic diversity. It is essential for evolution and speciation. If we talk about examples, so there are two examples. One is the single nucleotide polymorphism. It is the most common type. It involves what? It involves the change in single base pairs. And then is the variable number of tandem repeats, that is VNTRs. It is variation in the number of repeating DNA sequence. So this is the significance of polymorphism. Now we come to translation. If we define what is translation, so it is the process of polymerization of amino acid to form what? To form polypeptide. The order and sequence of amino acid are defined by sequence of bases in mRNA. The amino acid are joined by a bond which is known as peptide bond. So here simply they have asked what is translation, and we have defined translation. So it is a process where polymerization of amino acid occurs to form what? To form a polypeptide. Now, this order and sequence of amino acid are defined by what? These are defined by the sequence of bases that are present on mRNA, and these amino acids are joined by a bond which is known as peptide bond. What are the different steps of translation? So first step is charging of tRNA, that is also known called amino acylation. So this formation of peptide bond will require energy, and because of this, these amino acids are activated in first step. So what will happen with the help of amino acyl tRNA synthetase? It is an enzyme. This amino acids are this transfer RNA are activated. So what happened when this amino acid got attached to its tRNA? We say it is charged tRNA or amino acyl tRNA. Next step is what? Next step is okay, so we are talking about charging of tRNA only. So this activation of amino acid, these what happened in this? Amino acids are linked to their cognate tRNA, and this activation process is carried out in the presence of ATP. Activation process is also called what? Charging of tRNA or amino acylation of tRNA. Now, next is initiation. When the small subunit of ribosome encounters an mRNA, the process of translation of mRNA to protein begins. The small ribosomal subunit binds to mRNA at the start codon, which is AUG. The initiated tRNA, which is carrying this methionine, because we know that the start codon AUG also initiate the translation as well as its code for methionine, recognize start codon and binds to it. The larger subunit, ribosomal subunit, then bind to this complex, forming the complete initiation complex. So what is happening in this process? So simply what happened after this, amino acylation step, when this tRNA is charged, means tRNA has its amino acid. So what happened in now initiation step? This small subunit of ribosome will go and bind to the mRNA at start codon. Okay. Now what will happen? This initiated tRNA, which has this methionine amino acid, will come and bind to the start codon. Okay. This initiated recognizes start codon and binds to it. Now what will happen? Now this large ribosomal subunit also will come and bind to this complex, and it forms a complete initiation complex. Now what will happen after this initiation? There will be elongation step where complexes of amino acid which is linked to this their respective tRNA molecules bind to the mRNA. And how this binding will occur? This binding will occur via complementary base pairing between what? Between the tRNA anticodon and the appropriate mRNA codon. This ribosome will move along the mRNA codon by codon, and amino acids are sequentially added to growing polypeptide chain. DNA information, which is now represented by mRNA, is now translated into specific polypeptide sequence. So here what happened? We know this complexes of amino acid linked to the tRNA molecule will bind to the mRNA. This complementary base pairing occur between this tRNA anticodon loop, okay, the nucleotide which is present on the tRNA anticodon loop, and appropriate mRNA codon. This ribosome will move along the mRNA codon by codon, okay, and this amino acid is sequentially added to the growing polypeptide. And at last what will happen? The whole will be translated into polypeptide sequence. Then after elongation, there will be a step of termination when the ribosome reaches stop codon, which could be anything UAA, UAG or UGA on the mRNA. They don't have any tRNA to bind. Now, a release factor will bind to the stop codon. This will triggers what? This will triggers the hydrolysis of the bond that is established between the polypeptide and the tRNA. This hydrolysis allowed to release the completed polypeptide. After this, even the ribosomal subunit will dissociate from the mRNA. So here we can see what happened. Once this ribosome will reach the stop codon, okay, there is no tRNA which is going to come and bind now. So now what will happen? A release factor will come and bind to the stop codon. Here we can see it came and bind to the stop codon, which could be anything UAG, UA or UGA. Now what will happen? This will trigger the hydrolysis of the bond between polypeptide and the tRNA. Okay, so we can see the hydrolysis of polypeptide and tRNA happen, and what happened? This polypeptide is now free to go. After this, the ribosomal subunit dissociates. So this is the translation process. Now, last was bioinformatives. What is bioinformatives? So if we will talk about the definition of the bioinformatives, it is an application of computational and statistical techniques to molecular biology. What's the main purpose of it? It manages and analyzes the biological data, solves practical problem in biological research, create and maintain the biological databases. So what is actually bioinformatics? It is a field, interdisciplinary field in which we use the computational and statistical techniques to molecular biology. Its main purpose is that we manage and analyze the biological data, we solve the practical problems in biological research, and it create and maintain the biological databases. We have a lot of nowadays biological databases. Now, if we talk about historical context, so this emerged after the Human Genome Project, okay, developed to handle large amount of genomic data. Its key function is the data storage and management, development of the analysis tool, statistical analysis and all. So these are the historical context and the key functions of bioinformatives. Then if we talk about the importance of bioinformatives, it manages vast biological information, enables easy access for scientists, it facilitates the data interpretation, supports the future research. So these are some importance of bioinformatives. So we are done with today's class. Thank you.